Welcome to the Wright Science Daily Revision Boost. In today's Biology Blast, we're going to look at genetic engineering and biotechnology. So when we're talking about genetic engineering, we are talking about altering an organism's genome by selecting genes that create a certain characteristic. So we're changing both the genotype and the phenotype. Some examples of genetically engineered organisms: we've got cotton to increase its crop yield, corn to produce toxins that kill the insects that would normally damage the crop, and bacteria to produce drugs like insulin. And the way that we actually do this is we're going to use foreign genes, and these are ones that are taken from one organism that show the desired characteristic, and then we insert them into the plant or animal cells at an early stage of development. For those of you doing GCSE biology, you do need to know the risks here. So the key thing is that we don't know the long-term effects. Some possible negative effects may be health problems as we're introducing a new allergen into our food, and we could also potentially see cross-pollination with wild plants, which could disrupt the balance of the ecosystem. We also have those people who view it as unethical because science is interfering with nature. If you're doing the higher tier, either combined or biology, then we need to know how to produce a genetically engineered organism. So there are three key steps. Firstly, we identify the genes that code for our desired characteristic. Then we remove the gene from our donor organism. And finally, we insert the gene into our host organism. So the diagram on the right gives you a more in-depth description of this, but do remember that we use these restriction enzymes to cut the gene, and then we use DNA ligase to join that gene onto our actual plasmid. So the plasmid acts as a vector then to take it from the actual original DNA and insert it into our new cell. So we've got three enzymes that are actually involved here. The restriction enzymes, which cut the donor DNA at a specific base sequence, and we use that same restriction enzyme to cut the bacterial plasmid DNA, because that gives us complementary sticky ends. We then use a ligase enzyme to join the donor gene to the plasmid because it joins the sticky ends by forming a bond, and then complementary base pairing occurs through the action of another enzyme, which you luckily don't need to know the name of for GCSE. Once we've actually carried out this process, we need to check that the gene has been transferred, and the way we do this is through the use of marker genes. Now, these will either be antibiotic resistance genes or fluorescence genes. If it's an antibiotic resistance gene, we insert the marker gene into the plasmid at the same time that we insert the gene for our desired characteristic. We transfer the bacteria to an agar plate that contains the antibiotic, incubate it, give it time to grow, and then the colonies that are present on that agar must have the genes present because they're resistant to the antibiotic. Our second option for a marker gene is the fluorescent marker gene. So we again insert the marker into the plasmid at the same time that we insert our desired characteristic gene. We transfer the bacteria to an agar plate, incubate it, give it time to grow, and then to check the results we shine a UV light onto it, and any of the colonies that are glowing have our genes. So what we can then do is identify the colonies with our desired gene and then use those to build up very large numbers of our transgenic bacteria. The next thing we need to know is about how we use biotechnology in farming. So biotechnology is the use of biological processes or living organisms to manufacture products. And a good example of this are the genetically modified organisms. Two key types of GM crop that we have: golden rice, which has a gene from daffodil placed into the rice plants, and that means the rice produces beta-carotene, which our body uses to make vitamin A to avoid blindness. And the second one, BT corn, which takes the gene from the bacteria Bacillus thuringiensis and inserts it into a maize plant, and that means that it codes for a protein that's poisonous to insects to avoid the crop damage. For those of you doing a higher tier paper, remember a vector is the thing that's used to transfer foreign DNA to an organism. It could be a bacteria, a virus, or a plasmid. And the diagram at the bottom there just shows you how this is actually carried out in order to make our genetically modified organism. In today's chemistry check, it's a GCSE chemistry higher tier only section looking at the contact process and making ethanol. First one, the contact process, is a 3-stage process that we use to make sulfuric acid. 3 raw materials are needed: sulfur, air and water. So what we're going to do is have a little look at those 3 stages, but there's a diagram on the right that gives you a nice summary that you can learn. Stage 1 of our contact process is where we burn sulfur in air to produce sulfur dioxide. So you've got the balance symbol equation there, which you should learn. Stage 2 is where sulfur dioxide and oxygen react to produce sulfur trioxide, SO3. And again, you've got the balance symbol equation there, and this one is a reversible reaction. So we need key conditions for this. We use a pressure of 2 atmospheres, 450 degrees Celsius, and a vanadium oxide catalyst. The final stage, stage 3, is where sulfur trioxide is converted to sulfuric acid through the addition of water. Now, the key thing to remember here is stage 3 is actually very exothermic. So if we were to carry it out just as a single step, we'd get a very dangerous acidic mist produced. So we break it up into 2 stages. First one, sulfur trioxide is passed through concentrated sulfuric acid to make a substance called oleum, and then the oleum is added to water to make a large volume of concentrated sulfuric acid. When we consider why we've selected those conditions for the stage 2 reaction, because the equilibrium position is far to the right already, then we don't need high pressures, so we use the 2 atmospheres just to move the gases through the converter. The forward reaction is exothermic, which means our backward reaction is endothermic. We know a high equilibrium yield is favoured by low temperatures, but we use our compromise between yield and reaction rate, and therefore we're going to use the 450. It's also worth remembering that vanadium oxide, the catalyst, only works above 380 degrees Celsius, so we couldn't pick a temperature lower than that anyway. The second thing we're going to look at today is how we make ethanol. So we've got 2 different methods for doing this. The first one uses renewable raw materials, which we can replace as they're used so they don't run out. So these would be things like plants. And the way we do this is that ethanol is made in a fermentation reaction. So yeast contains enzymes that convert glucose into carbon dioxide and ethanol, and the glucose that we're using comes from plants. So I've given you the word and balance symbol equation there for the reaction. When we're considering the conditions for this fermentation reaction, if the temperature is too low, the yeast cells will become inactive. If the temperature is too high, the enzymes become denatured. So we normally carry out fermentation at 35 degrees Celsius and at normal atmospheric pressure. The second way we can make ethanol is by using non-renewable raw materials. Now the downside of this is that we're using them faster than they can be replaced, so they will run out eventually. And the one we're talking about here is ethene, which comes from crude oil. So ethene can be hydrated to make the ethanol. So to hydrate it, we're adding water in the form of steam. Again, you've got the word and balance symbol equation there. In order to carry out this reaction, the temperature is 300 degrees Celsius, 60 atmospheres of pressure, and we need a phosphoric acid catalyst. The last thing we need to be able to do is to compare those 2 different techniques for making ethanol and then decide which would be best. So in the table I've given you a summary about these key features: the cost of the raw materials, the conditions, energy requirements, the rate of the reaction, percentage yield, and the purity of the product. So make sure you've learnt a few of those, if not all of them, so that if they ask you which one would be the best process to use in a given scenario, you can justify your answer. In today's physics prep, we're going to look at insulation and efficiency. So the first thing we need to think about is when it's cold, we use heating in our homes to keep them warm. And if we turn the heating off, then the house cools down because the energy is transferred to the surroundings via the walls, the windows, etc. Now, we've got 2 types of wall that we find in houses in the UK. You either have a solid brick wall, which tend to be in the much older houses, or a cavity wall, which means there's a gap between the 2 walls of brick, which is usually filled with an insulating material. And the rate at which the energy is transferred through the walls will depend upon their thickness, the material they're made of, and the temperature difference between the inside and the outside. A measurement we use to determine how quickly the energy will be transferred through a material is the thermal conductivity. So the thermal conductivity of a material tells you the rate at which it transfers energy through an area of 1 meter squared, a thickness of 1 meter, with a temperature difference of 1 degree Celsius. The next thing we're going to look at is the efficiency. So the equation there you do need to memorise, they're not necessarily going to give it to you on the exam. So the efficiency is the useful output energy divided by the input energy times 100 to give it as a percentage. And what we find is that an efficient device, so one with a higher percentage, is better at transferring energy between the stores that do the job we want. One way that we can represent these energy changes that occur within any given system is using a Sankey diagram. And the key thing to remember here is that the width of the arrow shows the amount of energy that's transferred. So you start off with one arrow on the left showing our input energy, and then on the right we've got our output stores of the different types that we've converted our energy into. So just make sure that the thickness of your actual arrows, so the width of them, does represent the amount of energy. So they usually give you a bit of graph paper if they want you to draw this. Use an appropriate scale. The final thing for our higher tier candidates is knowing how to increase the efficiency. And to do this we just need to reduce the wasted energy. So we can do it by insulating, using materials that reduce unwanted energy transfer, or using improved technology like changing out your traditional light bulbs for LED light bulbs. And remember that more efficient devices operate at a lower power, so therefore they use fuels more slowly. As always, if there's anything we've gone through in our daily booster that you're not 100% certain on, head on over to the playlist to watch a more detailed video covering those concepts. For biology, it's the B6 playlist, chemistry, C6 playlist, combined physics is P5, and separate GCSE physics is P7.